A very warm welcome to all of you. In today's lecture, we'll discuss about the role of energy cycle in physical education and sports. Starting with the introduction, exercise is one of the greatest energy stressors that an organism is likely to encounter. Like other stressors, severe exercise has the potential to disrupt homeostasis. Every cell requires a certain amount of energy for life-sustaining processes. Thus, any disruption of cellular energy homeostasis is potentially fatal for the cell. Fortunately, we have evolved several potent mechanisms to maintain cellular energy homeostasis. Nearly every change in the body's physiology during exercise is centered on maintaining cellular energy homeostasis. For example, the increases in ventilation and cardiac output are meant to increase oxygen delivery to the mitochondria in active tissues, where oxygen serves as the final electron acceptor in the electron transport chain. The importance of energy metabolism during exercise is also illustrated by the fact that many of the possible causes of fatigue are related to cellular energy metabolism. The energy that allows us to perform life-sustaining processes, accomplish work, grow, and maintain body temperature ultimately comes from our foodstuffs. However, the energy contained in our foodstuffs is not readily accessible as a source of energy for the energy requiring processes of the cell. The energy from our food must first be converted to a common energy currency, ATP or adenosine triphosphate. It is thus ATP that serves as the immediate energy source for muscular work. Because muscle ATP concentration are very low during exercise, the production of ATP sometimes at very high rate. It is of highest priority. If exercise physiology were a story, the plot would center on energy and work. It is thus of extreme importance for exercise physiology students to understand the basic principles and concepts related to exercise metabolism. By this time, students have already learned in their introductory biology, human physiology and exercise physiology courses about the following basic cellular metabolic processes: the ATP PC system, glycolysis, glycogenolysis, gluconeogenesis, lipolysis, beta oxidation and tricarboxylic acid cycle, and the electron transport chain. It is recommended that students review these processes before coming to lab. These processes will only be discussed briefly in order to help students understand the big picture of exercise metabolism. A complete review of these processes is beyond the scope of this lab. Today's laboratories experiments are meant to help students better understand those factors, for example, exercise, intensity, duration, etc., that determine what fuel sources and metabolic pathways are contributing to an exercise bout. Exercise as a metabolic stress. At rest, almost all of our energy is coming from aerobic metabolism and results in an oxygen consumption of around 3.5 milliliter per kg per minute or about 0.25 liter per minute in an average human. Assuming a caloric equivalent of 5 kilocalorie per liter, this means that at rest, most of us expend about 1 to 1.5 kilocalorie per liter. An average college age male can reach energy expenditures of 15 to 20 kilocalorie per minute at maximal exercise. Olympic rowers average 36 kilocalories per minute during a stimulated 2000 meter race, 5 to 7 minutes, and can attain energy expenditures up to 40 kilocalories per minute during training and competition. A 40-fold increase in energy expenditure above rest. Of course, these high rates of energy expenditures cannot be sustained for long periods of time. Among the highest recorded daily energy budgets to be maintained for more than a few days are for cyclists participating in the Tour de France, where daily energy expenditure was around 7,000 kilocalories per day for 22 days. This is almost 4.5 times the typical basal metabolic rate, that is 1600 kilocalories per day for these athletes. Clearly, exercise places extreme demands on the body's energy systems. For example, it has been demonstrated that flux through the citric acid cycle or Krebs cycle can increase over 100-fold during moderate to high intensity exercise. Sources of energy. The only direct source of energy for triggering muscular contraction and hence movement is adenosine triphosphate, or generally called ATP. ATP cannot be stored in the cells in large quantities. Therefore, it is in short supply in the muscle cells and it must be constantly recreated or resynthesized. This process of resynthesis of ATP after it has been broken down to adenosine diphosphate, that is ADP, in the process of triggering movement also requires energy. This most commonly used sources of this energy is creatine phosphate. In the final analysis, most energy fuel for this purpose is derived from carbohydrates and fats. With the use of energy from these sources, the resynthesis of ADP and ATP is accomplished through either of the three systems. These systems are the ATP PC system, the oxygen system, that is aerobics, and the lactic acid system, that is anaerobics. Ultimately, oxygen is needed for the release of all energy sources. The energy pathways of exercise. Muscle cells are unique with respect to their capacity to use oxygen. Which one of the resynthesis system or energy pathways used at any given time depends on several circumstances, including the amount of oxygen present at that time. For example, aerobic means muscular work in the presence of oxygen, while anaerobic employs in the absence of oxygen. The aerobic processes can use all the sources of energy listed in the above unit except creatine phosphate. The anaerobic system can only use glycogen. The ATP PC system is the simplest and most direct and uses only creatine phosphate. However, all three systems operate in a similar manner. When the energy resulting is used to reform or resynthesize ATP, there is a continuous cycle as fast as ATP is broken down to ADP during muscular work. It is being resynthesized from ADP by the energy liberated by one of the three systems mentioned above. Both the chemical or metabolic pathways, the lactic acid system and the oxygen system, operate through the process of glycolysis. This is the chemical breakdown of glycogen to pyruvic acid to produce energy for the resynthesis of ATP. As soon as there is sufficient oxygen in the cells for oxidation, aerobic glycolysis is employed and all the three sources of food, that are glucogen, glycerol and fatty acids, are broken down. When the demand of metabolic processes exceed the oxygen available, anaerobic glycolysis prevails. In this case, Glycogen is the only source of blood used. The ATP PC system. The simplest of these systems is the ATP PC system. Since it operates in the absence of oxygen without a buildup of lactic acid, the energy derived from this process is a result of the breakdown of phosphocreatine with phosphate and creatine as byproducts. However, phosphocreatine, like ATP, is in short supply and can soon be depleted after a short period of time, usually 1 minute or even less. It is like ATP in another way. It too has a phosphate group, and when its bonds are broken, a large amount of energy is released. Therefore, it can provide immediate powerful bursts of activity, but such activity must be followed by rest to replenish the phosphocreatine. Normally, rest period must be from 3 to 4 times longer than the duration of the activity until other systems can take over. The oxygen system or aerobic. In the process of aerobic glycolysis, glycogen is broken down to furnish the energy to replenish ATP. During this process, water and carbon dioxide are liberated to be transported to the elimination centers. As long as an adequate supply of oxygen is available for the combustion in the oxidation cycle, exercise can continue indefinitely because the triggerer of muscular contraction, ATP, is continuously being resynthesized within the minute power stations of the muscle tissue called mitochondria. In this process, there is little accumulation of lactic acid in the muscle tissue because in the aerobic processes of energy used to resynthesize ATP is derived from a breakdown of glycogen to pyruvic acid. Pyruvic acid then goes through chemical reactions in the mitochondria known as the Krebs cycle, where oxygen is used continuously for energy to reform ATP. In this manner, ATP can be replenished constantly without the accumulation of lactic acid, and the three foods can be broken down completely, yielding a large amount of ATP molecules. To further complicate the picture of energy release, neutral fats instead of glucose may be used in the aerobic glycolysis process to provide the needed energy for ATP resynthesis. Triglycerides are broken down into glycerol and fatty acids. Glycerol then enters the glycolytic pathway, and in the usual manner as glycogen is used for the purpose of resynthesis. Fatty acids, however, must go through the Krebs cycle for oxidation similar to pyruvic acids before they can be used for regeneration of ATP. The lactic acid system. For longer period of exercise without oxygen and too long to be handled by the ATP PC system, the lactic acid system using anaerobic glycolysis prevails. In this case, during the process of resynthesizing ATP, glycogen is broken down to pyruvic acid. Since sufficient oxygen is not available, anaerobic glycolysis continues to break down glycogen, but pyruvic acid spills over the tissues and accumulates as lactic acid. Lactic acid leads to fatigue shortly and eventually to exhaustion. There is something unique about the body's use of oxygen. While other body cells do not have the capacity to tolerate a deprivation of oxygen even for a short time, muscle cells do. Therefore, they are different because they have the capacity to tolerate a temporary deprivation and incur and oxygen dipped. When the intensity or duration of the exercise requires more energy than can be made available by the cardiovascular respiratory system, the muscular metabolism system achieves an anaerobic state so that exercise may continue for a period of time without sufficient oxygen. However, during such exercise, there is an accumulation of lactic acid which eventually results in muscular fatigue and exhaustion. Also, the amount of energy released by anaerobic glycolysis is approximately 20 times less than that produced by aerobic glycolysis. The process of converting glycogen into lactic acid is reversible. The major portion of glycogen used in anaerobic glycolysis during exercise can be resynthesized during rest by the liver, but this process takes time. During exercise and afterwards, the lactic acid diffuses out of the cells and into the bloodstream. Some of it may be temporarily neutralized by buffers in the blood and some is carried to other parts of the body removed from the exercising part. Smaller amounts may be excreted by the elimination centers. However, since the first two of these are temporary measures, eventually all lactic acid in the body must be oxidized. Much of it is carried to the liver for resynthesis into glycogen to be disturbed into the bloodstream once again. Foregoing explanation indicates why an aerobic activity can be carried on for a longer period of time than an anaerobic activity. The buildup of lactic acid is much less. However, in the anaerobic processes following a rest or during mild activity, the deficiency in oxygen can be overcome and the oxygen debt can be repaid through increased activity of cardiovascular respiratory system. In summary, under a crest load type of exercise, oxygen requirements of the muscle cells equal to the oxygen uptake, and the respiratory system effectively exchanges oxygen and carbon dioxide to meet the needs of the cell. Little lactic acid will be formed in the tissues because there is an adequate supply of oxygen in the mitochondria for oxidation in the Krebs cycle for aerobic processes. However, when the intensity of exercise is increased beyond the crest load, the demand for oxygen, which cannot be stored in the cells, is not transported by the circulatory system fast enough for aerobic processes to prevail alone. Pyruvic acid continues to be broken down through glycolysis to resynthesize ATP, but the lactic acid, which is not oxidated in the mitochondria, spills over into the cells and accumulates as lactic acids. Consequently, aerobic glycolysis becomes a secondary source of energy producing while anaerobic glycolysis predominates. Rasch and Burke suggest that both aerobic and anaerobic processes prevail during muscular activity, but one or the other generally predominates. Implication of the energy pathways. First, it is apparent that a trainee or a performer does not require an immediate supply of oxygen for movement. In the absence of oxygen, the energy for synthesis of ATP can be derived from anaerobic sources. Also, the ATP PC system has no involvement in the lactic acid system, but of course, it can be employed only for brief bursts of activity before period of rest in order to replenish phosphocreatine. For longer periods of activity in the absence of oxygen, the anaerobic lactic acid system prevails. However, If the activity extends beyond a few minutes and if the circulatory training effects are to be elected, the major energy source would be aerobic glycolysis, utilizing the oxygen transport system. Muscle glycogen depletion is possible cause of fatigue during exercise lasting over 90 minutes. Thus, sparing muscle glycogen would benefit individuals during endurance exercise. Exercise training tends to increase the use of fatty acids as a fuel source. Thus, at any given intensity, a trained individual has to rely less on glycogen, sparing their glycogen stores. There is some evidence that caffeine may spare glycogen stores by increasing reliance on fatty metabolism. However, there is also evidence that caffeine does not spare muscle glycogen. Carbohydrate ingestion during exercise also tends to spare glycogen by providing another source of glucose for glycolysis. The fatty acid used to produce energy by skeletal muscle are derived primarily from adipose tissue triglycerides and to a lesser extent from intramuscular triglycerides. It should be noted that triglycerides or TG must be broken down before they can be used as a fuel substrate. The degradation of TG into fatty acids and glycerol is called lipolysis. Inside the cells, lipolysis is catalyzed by the enzyme hormone sensitive lipase, that is HSL. A different lipase, lipoprotein lipase LPL, catalyzed the breakdown of TG in the circulation and allows adipose cells to take up fatty acids from these circulating TG. Once fatty acids have been taken up by adipose cells, they can be stored as TG. This is called lipogenesis. While both LPL and HSL are involved in triglyceride breakdown, they play very different roles in the fat metabolism. LPL tends to increase storage of fatty acids in adipose tissue, whereas HSL mobilizes fatty acids so they can be used as an energy source by their tissue. Summary of the storage and breakdown of carbohydrate and lipid fuel stores. With this, we come to an end to today's lecture. I hope this must have added to your concerns about this important topic. Thank you very much. Have a nice day.